Okay guys, now that we've gone through some information on microbial metabolism, we are now going to look at the effect of that metabolism. No cell can grow, no population can grow unless the metabolic abilities are there. So now we're going to look at bacterial growth. Okay. Now, first of all, there is a difference between the division of cells within eukaryotes and prokaryotes. And one of the differences is bacteria, which are prokaryote, they divide through binary fission, a mode of reproduction whereby everything is basically replicated and then you end up with two daughter cells. Okay? Slightly different than mitosis in the way that the DNA is replicated and so on. And what we can talk about is how long does it take from generation to generation when bacteria grow. Okay, we can talk about that with the human population, right? In the teens, a human female is capable of carrying a child to term, all right? And so here now, we're not talking about years anymore. We're talking about minutes, okay? So generation time is the time required for a cell or individual organism to divide or replicate. Now, this can be as fast as 20 minutes, 25 minutes in between generations. And for that quick a time, we could be talking about Staph aureus, which is 25 minutes, E. coli, which is 20 minutes. But some microbes grow much more slowly, and their generation time is on the order of maybe 12 hours. Now, this can be pretty relevant when it comes to diagnosing disease, right? If there's an organism that's causing a disease, then if we want to find out what it is and culture it in the incubator, well, you may be waiting a while if an organism grows that slowly. So classic examples of these might be Mycobacterium tuberculosis, which obviously causes tuberculosis, Treponema pallidum, excuse me, which causes syphilis. So you don't want to be waiting for a diagnosis based on a live culture that you're growing in the lab. You want some other way to test for these particular organisms that have such a long generation time. And we've already talked about one of those ways with the tuberculosis species, and that is do an acid fast stain, right? So the stains we've already talked about. All right, now if we were to think about a single cell that is undergoing division, we could talk about it's going, one cell divides into two cells. Two cells divide into 4 and 8 and 16 and 32, and it just continues to double from there. And before you know it, you're up into some really big numbers. And this would be a synchronous growth of a culture, right? They are all, these cells would all be in sync with one another as they divide, okay? And the population would go up in a stepwise fashion, okay? Versus asynchronous growth, where they are not in a stepwise fashion with one another. All right, so here we have an image of binary fission. I'm not expecting you to know the specific steps within binary fission, but definitely you should know the difference between binary fission and mitosis, just simply in the fact that one occurs in prokaryotes and the other occurs in eukaryotes. Okay, this image kind of shows you that stepwise fashion we would expect if a single cell goes through binary fission. We end up with 2, 4, 8, and so on. This graph over on the right indicates that with this number would go up very quickly, and for that reason we usually use a special logarithmic type of scale to measure this, because very soon you're up in the thousands starting from a single cell. Now this graph shows us more or less the stepwise fashion, okay, with the blue line here, that we would expect from a kind of growth that is synchronous, okay? There's a stepwise fashion going on versus an asynchronous or non-synchronous, another way of saying it, without synchrony growth that we might have otherwise. Now, let's consider, we see this image here on the right? So here someone is taking a sample, let's say of a pure culture from a source, and they're putting it into a broth tube. Now, if you pick a sample, the chances that this sample, all of those cells are in the same life stage is pretty slim, okay? So the chances are that as we pass a sample into a tube like this, unless we've done something pretty special to make sure they're all at the same life stage, I would expect that they are different times away from dividing, okay? So a good analogy might be, okay, take everybody in our class, okay? A couple people in the icebreaker assignment said they were in their 40s and 50s. I'm in my 50s. We have some people that are in their teens, okay? So we have people in lots of different life stages, right? Some of us, are past reproductive age. Some of us are in their reproductive age, right? So we have to think of it the same way, okay? Cells like that are in the varying stages of their life cycle would show something of a non or asynchronous growth. It would be a smoother curve because these various individuals would be undergoing a reproduction at different times, okay, but still would lend to a growing population of those cells. All right, so one very routine thing to do in a microbiology laboratory is to determine the growth rate of organisms, okay? If we were to take, again, we see on this image at the bottom right, if we were to take some organisms with a loop, okay, with a sterile instrument, and introduce them into this tube, and put them in an incubator, let's say 37 degrees, well, those cells have now been introduced to a brand new environment, okay? They may have been in their original environment kind of just chilling, right? I mean, they may not have had a great amount of nutrients. They're fighting for space. Let's say they were in an older tube where a lot of cells are growing, and now you introduce them into a brand new fresh tube of broth, okay? Well, it's going to take for some time for them to get acquainted and start gearing up their metabolism for the use of nutrients, sugar, and things like that within that tube. There will be a lag phase. There will be a time during which there is not a lot of growth. They're just starting to gear up their metabolism for growth. But then, once they get familiar with that environment, and those metabolic processes are in full gear, they're going to grow very quickly. 1 becomes 2, becomes 4, becomes 8. They're not quite in that stepwise fashion, probably, because they are at different life stages, but this would very quickly grow in the population of those particular organisms. Eventually, there would be a limit, okay? So here we have a graph showing that lag phase, right? So we introduce a certain number of cells. those cells would be under some stress getting used to that environment, and then ultimately we see that this curve goes up. We have the log phase or the exponential phase where there's exponential growth. Now eventually these organisms are all going to be competing for the nutrients in that tube. Okay, so there's a certain amount of sugars, proteins, lipids, and so on in this broth. They're going to start to compete for space within that tube and nutrients within that tube. And for that reason, there will be a certain amount of cells that are going to start to succumb to the environment. There is going to be a certain amount of cells that will die, okay? But we'll have a stationary phase where the population remains static, all right? Eventually, the sheer concentration of waste within that tube, within that one tube, will contribute to the poor health of many of the cells in that tube. And there'll be a decline phase. There will still be living cells in there and still cells that are replicating, going through a reproduction process, okay? But this population will crash. Sometimes we call that a death phase as well because of that movement downward, okay? So here we have another image kind of showing that idea and the fact that there's going to be some dead cells in these tubes as we go on into the stationary and the death phase, okay? Here we have an image that kind of shows some research somebody had done, and they're showing different growth rates, but they all look like a growth curve like we saw before, right? You should be able to recognize a lag phase. All of these curves have a lag phase. Some of those lag phases go longer than others. They're showing a log or exponential phase, and some of those go up a little faster than others. Stationary phase. This particular curve here to the very right, it's still in its stationary phase when they terminated their experiment, right? So all of these phases can go for different lengths depending on lots of criteria. What is the cell that we're working with? What is the species? What kind of nutrients did you give each of these cells? Because we know from our lab that some organisms grow well on certain sugar sources, some do not. I would say that these particular, let's put it in red here, this particular growth curve here tells me that the organism with indicated by a triangle symbol here seems to be very happy, right, in this environment, whatever it is. It's going up very quickly, and the fact that the stationary phase is short only means that it's really using up the nutrients quickly and getting to a point where it's reached its limit faster than this other one over on the right, okay? Now, there are different methods for culturing, and the one method we've used and talked about the most involves a plate or tube in lab. These are batch methods for culture. There is a limitation inside of a tube and inside of those plates that we've talked about for the nutrients that are in there, okay? This would be referred to as a batch method, okay? A method by which a set of cells or a culture is grown with a limited level of nutrients available, okay? There's a possibility that those nutrients will become scarce and that there will be a limit to the population that can grow on that scarce level of nutrients. The other possibility would be a continuous culture, a situation where the nutrient level does not decrease, nor is there a limit. So basically, this would require us to replace nutrients as they're used. This would be a continuous culture. All right. So all of the methods with tubes and plates, these are all batch methods for culturing where the nutrient level is limited. So I thought I'd show you a few real examples of these kinds of culturing methods. So here are some flasks, somewhat like I used in my master's, actually, when I was doing my master's degree, where I grew cyanobacterium, a cyanobacteria that is green, obviously, and it's using the sun's rays, the light. It's a phototroph to get its energy. But the one thing that is not done here is there is no replacement of the nutrients that are in this flask. So this would be a batch method of culture. Sometimes for culture like that, we would bubble in some air because it's natural for there to be a certain amount of bubbled air in there, like with the waves where we see these cyanobacteria on the shore. But always we would be careful to keep things sterile, right? So we'd have a charcoal filter to introduce air, but not nutrients. So it's a batch method still. Here we have a picture from a sewage treatment plant. All right, so here we would have the natural source of nutrients would be human waste, right? And that human waste just keeps on coming, right? So here we would have a continuous culture. We'd have a constant adding of the nutrients for these bacteria that help to break down our own waste. Here we have a more sophisticated vessel where they would actually control for the pH in these vessels. But in this particular example, I don't see any place where they would add nutrients. So still a batch method. All right, here we have a little more sophisticated continuous cultivation system. So in a laboratory, you will see pumps that are pumping materials into a vessel. This cultivation tank would be where the organisms are growing. We have all sorts of things that can be really controlling this in a laboratory. Control panels. We could be controlling for the air that's circulated within this cultivation tank here. But the one thing that I can see is that this constant pump seems to be adding a nutrient source. So, continuous. And here we can see that kind of set up in a laboratory. Okay, the lab, one of the labs down my hall, they work with Pseudomonas, and Pseudomonas seems to grow on surfaces within catheters and other places like that where fluid is moving. Okay, so here they have tubes that are all set up to a pump. We have a control center here that's controlling how fast fluid moves through those tubes, and all of these tubes that you see here, so here in green you see this tube, right? All of these tubes have bacteria flowing through them, all right? In this particular case, this would be a continuous situation where they keep adding nutrients. Okay, finally, one of the things we want to be able to do is actually measure growth. How is it that we come up with this graph, this growth curve that we went over? Lag, log, stationary, and death phase. You want to know those phases. How are we coming up with the actual numbers to make that graph is the question. One of the ways we can do this is by directly counting the number of cells that we have in a culture. So let's say that we inoculate a broth tube or a flask, some kind of vessel with nutrients within it, okay? We could take a sample every hour, every 2 hours, you pick your timing, right? And go to a microscope and actually measure the direct, count them, the number of cells that are present in a certain volume of that liquid. Okay, so this would just be a situation where we're using a microscope to do this. Now, this direct count, if it's with a naked eye that we're doing this and there's no staining involved, may be inaccurate. You may be counting both live cells and dead cells at that point. So there's no way of knowing how many cells are still alive in that culture. Okay? But this is one way to approach it. Some types of direct counts will have a staining involved, a fluorescent stain that will show us with certain colors the cells that are alive versus the cells that are not alive. Okay, so there are ways around the alive versus not alive situation there. But there is another way of finding out how many viable cells are present, how many living cells are present that will give rise to new cells. And one of them we've talked about in lab. Okay, this would involve possibly doing a dilution, set of dilutions within tubes, and then plating, either with a spread plate or a pour plate, plating these cells out and incubating those plates. Now, the only cells on those plates that will give us colonies that we could see later would be living cells. So, the spread plates, the dilutions with spread plates that we've been talking about in lab, viable counts are what those are, okay? So this will give us an idea, a more clear idea of how many cells are alive there. Now, there are other optical techniques that we can use. If we were to take samples from each of these tubes, okay, at different times, we would find throughout a, let's say we inoculate a tube, then we go in and we take a sample, and then another 2 hours later we take another sample, and so on, just like we did up here in the first example. And then use a special instrument to see how much light would pass through that sample. Okay, like with a spectrophotometer. Okay, some of you might be familiar with a spec like this from a chemistry lab. Okay, but in this case, we're using light and seeing how much light actually passes through the sample. If a lot passes through, we know there's not a lot of cells there. If a lot of light is actually directed elsewhere and absorbed by the cells that are in there, then we might expect that there are more cells in that tube. So these optical techniques can be used. It's more of an indirect measure of the cells that are in there based on the amount of light that can pass through the tube. Now, there are other optical techniques, much more sophisticated techniques that they use in hospitals and other laboratories that are automated, and they can actually measure in a much more sophisticated way. But in the end, to make a growth curve like that, we need to count the cells. So we have direct, where we can get both living and non-living, viable counts like we see with those dilutions and plating on the plates. And then we have optical techniques where we can use light to show us how much light gets through or is actually absorbed by cells in there. Okay, we will catch up next time with some of the different parameters that affect the growth of cells.